Okay, last call before departure. Do we have Jack Dodson with us today? If yes, could you please make yourself known? Otherwise, we're gonna kickstart this session. So, hello everyone. My name is Baptiste Kouvidichan, and we have Nicolas Gallagher with us today. We will be chairing the session. And so, in the spirit of reconciliation, it's a custom in Australia to start a gathering or a meeting by a quick acknowledgement of country, which I'm going to do now. So we're gathering today on the land of the Turrbal and Yugara people, who are the traditional custodian of the land. And I would like to pay my respects to the elders past, present, and emerging. Without further ado, we'll start with our first speaker of the day, Yohan Yee, who's coming from McGill University. Thank you.
Okay, hi everyone. So thanks for that introduction. I want to start by acknowledging that many people have been involved in this work that I'm going to talk about, and they're all listed here. So everything I talk about is in a preprint that's on bioRxiv, and I also have a poster that's up today and tomorrow. So if you want a copy of these slides, if you want more info on the poster, on the manuscript, feel free to take a picture of this QR code. I'll leave it up for a few slides, but let's begin. So just a quick disclosure, one of the authors involved in this work has some disclosures, but that's unrelated to the study. Okay, so I'm sure that many of you are very familiar with the Allen Human Brain Atlas, but to really quickly summarize, it's a really popular data set that describes gene expression in the human brain. Basically, the Allen Institute used microarray-based methods to capture genome-wide gene expression, and for this they took tissue samples, 3,702 tissue samples across the postmortem brains of 6 donors. So it's a widely used resource in neuroimaging, and that's because the locations of these tissue samples are provided in relation to an MNI space template. And this has resulted in the Allen Human Brain Atlas being used to study the relationship of gene expression to brain structure, in this case to brain function, the transcriptomic correlates of disease. And it's so popular now that there are practical guides and toolboxes that standardize these comparisons between spatial gene expression and brain phenotypes. So these studies are what I call imaging transcriptomics. And the number of imaging transcriptomic studies have rapidly grown over the past decade. So all this data here that you see is from PubMed. So I want to emphasize that imaging transcriptomic studies rely on gene expression data that's sampled from points within donor brains, being accurately placed within an anatomical template over which neuroimaging findings are represented. So the Allen Institute originally provided locations in relation to the MNI nonlinear 6th generation asymmetric template, which I'll call the original mapping, or the original coordinates, and this was done through image registrations of donor brains to this template. So it turns out that for 4 of the 6 donor brains, only an affine alignment was done. So shown here in the bottom right are an affine aligned subject brain to this template. And to see how an affine only alignment can be an issue, I'm going to take this template brain shape as represented by these contours, and I'm going to overlay this on this affine aligned subject brain. So right away you can see how an incomplete alignment can result in tissue samples from subject brains not being accurately placed in the right anatomical location in the template. So another good example is from this other subject, where in this axial slice you can see how cerebellar misalignment here can really affect point-to-point correspondence between the subject and the template brain image. So these original coordinates were made available around 2012 or so, and soon after I think people recognized this issue of inaccurate localization. And so in 2014, Chris Gorgolewski provided an updated set of coordinates, so this is used in the Abidjan package. And this was done through nonlinear registrations of these donor brains to the same brain template. And so he did this in what he calls the AllenInf software package. So I'll call these the AllenInf coordinates. And most recently, Gabriel Deveni re-registered these donor brains to a newer MNI nonlinear 2009C symmetric template But here he used both T1 weighted data and T2 weighted data in a joint optimization. So this happened at the Cerebral Imaging Center in Montreal, so I'll call this the CIC coordinates. Until now, there really hasn't been a formal comparison of these three different coordinate sets. And so we began by asking, how accurate are these coordinates in really describing the true locations of where these tissue samples came from in relation to these templates? So the first thing we did was we visually inspected the true locations of these tissue samples relative to the subject MRI, and that's shown here on the very left. We can do this because we know the true location relative to the donor brain, and we also know the structure from which these tissue samples were taken from, because this was recorded by the expert neuroanatomist at the time of dissection. We can compare this to each of the three coordinate-based locations relative to their appropriate template. So as you can see in this example, a sample here taken from the cerebellum is incorrectly placed in the cortex by original and Allen-Inf coordinates, but here it shows up in the cerebellum using CIC coordinates. We can quantify this mislocation between core structures, such as the cortex, subcortex, brainstem, cerebellum, and the white matter. So highlighted here are samples that are truly taken from the cortex. And what we're looking at are the mislocation, or the proportion of samples misclassified. So these are samples that are actually placed in other structures other than the cortex. And as you can see, about 12 to 13% of cortical samples are misclassified when using CIC coordinates, but this is much lower than original and Allen-Inf coordinates, where up to over 30% of cortical samples are placed in other structures like the cerebellum or the white matter. And this is the case also for samples taken from the subcortex, brainstem, cerebellum. And CIC coordinates only really do worse for non-gray matter tissue samples, but there are only about 20 of these in the Allen Human Brain Atlas. So beyond this neuroanatomical placement of coordinates, we also looked at image correlations using these subject and template brains. So to kind of show you how this works, I'm showing you images of unaligned and aligned subject and template brains at the top left and bottom left here. And I've kind of downsampled them so you can see the voxels better. So imagine if these red boxes were the tissue samples, the locations of tissue samples, and the colors within are the intensity of the image under the tissue sample location. So you can take these intensity values under the tissue sample locations in the template image, plot that on the horizontal axis, take the intensity value of the subject image, plot that on the vertical axis, and if these images were not aligned, you'd expect these points to be kind of scattered all over the place, but if they were aligned, even if the intensities were a totally different scale, you'd expect to find a higher correlation. And so using this test, we computed these correlations separately for each set of coordinates. So here I'm showing you what you find with one donor brain. And we find that CIC coordinates provide the best correlation in this case. This is for a single donor brain. I'll represent this correlation using a bar plot with confidence intervals here. They're obtained through bootstrapping. And looking at all other donor brains, we find that CIC coordinates were generally much better than the other coordinate sets. These correlations were computed using T1 weighted subject and template images, but we also have T2 weighted data for the subjects and the template. So even when we use that, we find that CIC coordinates are most optimal. So far I've described two tests in which CIC coordinates perform best. We actually carried out 7 independent tests of accuracy under different processing, analytical choices, all of which pointed to CIC coordinates being most optimal. I won't describe that here in the interest of time, but check out the manuscript, come to the poster. I'm happy to describe that more in detail. But hopefully at this point, I've convinced you that CIC coordinates are more optimal than the other coordinates. But the question is, does that choice actually matter in a typical imaging transcriptomics study? So to answer this, we first turned to this eNeuro study here that looked at a meta-analytic map of PET findings in Alzheimer's disease. And here the authors essentially took spatial gene expression, correlated it with this phenotype that you see on the right, and determined rank differences. So we reran the exact same analyses, but we only changed the coordinates each time. So we changed it from original to Allen to CIC coordinates. So comparing original and CIC coordinates, I've plotted the rank of genes here so that each gene is a point. The rank obtained using CAC coordinates is plotted on the x-axis. The rank obtained using original coordinates is on the y-axis. And you can clearly see that there are genes that are almost top ranked with original coordinates, but bottom ranked with CIC coordinates. So that's points at the top left that are in red. And then there are also genes that are on the opposite side. I'm going to rotate this plot a bit, make it a bit more interpretable. Essentially here now the horizontal axis is rank difference. And you can see that the rank difference when you use different coordinates is very high. This is the same case when you compare CIC to ALAN-INF coordinates, and we find that there are many genes both on the left and the right side. So we can quantify these biases by projecting these points into a horizontal plane, which gives us distributions of these rank differences. And here you can see whether you use original or ALAN-INF coordinates compared to CIC coordinates, about 20% of genes have an absolute rank difference of over 4,000, which is a surprisingly high number. This rank differences would be measured in a single study. We wanted to know if this was generalizable. So to know this, we turned to NeuroQuery. We took about 6 meta-analytic brain maps from NeuroQuery, and we repeated this comparison for each different NeuroQuery map. So NeuroQuery takes in terms, predicts where brain imaging findings will be located. And here, as you can see, there's an example for the term auditory where predicted findings are localized to the temporal lobe as expected. So here, the rank differences that you see when you use the NeuroQuery map corresponding to the term auditory, you can see similar distributions in the shapes and the sort of the large spread in rank differences when you use original or ALAN-INF coordinates. And using this, we then quantified bias and downstream enrichment analyses. So I won't go too much into detail, but basically each map here, we took the gene ranks obtained using these different coordinate sets, ran an enrichment analysis, identified the gene ontology terms, pathways that were implicated, and just like we did for the gene ranks, we can plot the difference in the enrichment values we get for each gene ontology term or pathway term when using original or ALAN-INF coordinates. So each point here is a NeuroQuery map, and the enrichment quantified the enrichment difference is plotted on the x-axis. So the terms you see here are highly impacted. This means that for most NeuroQuery maps, there's a really large enrichment difference that's significantly different from 0 if you use the previous inaccurate coordinates. And so across all terms, we can look at which ones have biases that are significant across all NeuroQuery maps, and we found that for about 11% of gene ontology terms, we found significant false positives when using original coordinates. 21% of terms are false positives when using ALAN-INF coordinates, and around 43% of pathway terms from the pathways data set were false positives, so that was like the worst case. So from this, we can conclude that coordinates that you use when working with the Allen Human Brain Atlas can actually make a big impact in your downstream gene ontology and pathway terms that you kind of find when you do an imaging transcriptomics study. So to finally summarize, we found that CIC coordinates were most accurate in placing tissue samples within their correct neuroanatomical locations in relation to an MNI space template, and that coordinates matter. So if you use older, less accurate coordinates, you can really get different gene sets, different genes that are identified, and different sort of pathway terms, GO terms, gene ontology terms that you might falsely identify. So with that, happy to take any questions. I'll put up the QR code again if you want the link to the preprint. And I'd like to thank everyone at the Computational Brain Anatomy Lab that's shown here in this picture.
Is this? Yeah, a lovely presentation, lovely study, and as an anatomist, I can only say, of course it matters. I mean, if you've got a probe that's taken from the cerebellum, but you're calling it cerebral cortex, anyway.
It matters, yeah.
Yeah. Questions. Given these discrepancies, how do we interpret previous imaging transcriptomic findings?
Oof, that's a difficult question. Short of asking you all to redo every single study, I think we should just take every kind of conclusion with caution. I mean, at the end of the day, imaging transcriptomic studies are correlative. There's a lot more issues than just mislocation. Often you're taking normative data acquired in other subjects, comparing it to a totally different data set. We know that there's a big sex bias in these donors. 5 of them are male, 1 are female. So there's a lot of questions that I think that come up that I would say you should use caution when interpreting these findings. And it's not just because of these coordinates.
Right, but it's just an added
Exactly.
point on that one could discuss.
Yeah.
Okay.
I think we may have to move on, but
Yes.
waste of time. But come and talk to Yohan after if you have more questions. Please do. Thank you very much, Yohan.
Thank you.